Chen from Harvard University.
Thanks. Thank you for inviting me. I'm going to talk about information in the prebiotic world. So this is going to be a little bit different. And specifically, I'm going to talk about how the mechanisms of synthesis in the prebiotic world would have influenced the information in the genome. So one of my favorite writings on this area of synthetic biology and origins of life is from the 1600s. And this Flemish chemist said, if you press a piece of underwear soiled with sweat together with some wheat in an open-mouthed jar, after about 21 days, the odor changes, and the ferment coming out of the underwear changes the wheat into mice. So this is kind of an early proposal for a synthetic biological experiment. And our understanding has changed a little bit since then. This is a reflection of the modern understanding of the timeline for the origin of life, starting from the formation of the Earth and going to the diversification of life as we know it today. So what I'm going to focus on is this transition between prebiotic chemistry, so the Miller-Urey type of experiments that produce small molecule monomers, to the production of polymers. So we would go from things like, say, amino acids and nucleobases and nucleotides through some polymerization chemistry. Several examples of this type of prebiotic reaction have been shown, for example, on mineral clays or during heating and drying cycles of lipids, taking these monomers to polymers. And once you have polymers containing more than one type of monomer, then you have information in the sequence. So what happens to information over time? I think we can kind of intuitively say that information or the complexity of the sequence increases over time or has increased. This raises two questions. The first question is, how much information did we start with? So as soon as we got something that could carry information, how much information could there have been? And the second question is, were there any mechanisms that drove this increase in complexity or information? So starting with the first question, how much information can you carry in a polymer? This question was actually first addressed by Manfred Eigen in 1971. And the way he posed this problem is that genomes are constantly mutating. In other words, the synthetic mechanisms are not perfect, and so you're constantly making mistakes. So if you have a particularly good replicator sequence, let's call it the master sequence, it produces both master copies, so perfect copies of itself, as well as mutant copies, so mistakes. So depending on what the error rate is, if the genome is relatively short, then you can make a sustainable number of perfect copies, and occasionally you make some mutants. But if the genome is relatively long, at the same error rate, you'll make primarily mutant copies, and the information of the master sequence will be degraded over time. So Eigen formalized this into the error threshold, and the idea is that at a given mutation rate, the fittest sequence cannot persist if the genome is longer than a critical length. So it's kind of a phase transition. And this critical length is proportional to the log of the fitness of the master sequence, so the relative fitness of the master over the mutants, divided by the mutation rate. So this inverse proportion is particularly important. So to back up a little bit, the model that Eigen solved was for basically the simplest possible replication, which would be a single kinetic step. So like x goes to 2x in one step. But while this might be a good approximation in some situations, we can still try to inject some more realism into this model. So in other words, we wanted to try to determine what the error threshold would have been in a realistic prebiotic scenario. So what are the mechanistic details that we want to include? The first is that polymerization stalls after mistakes. So if you have, let's say, a template sequence and a primer that's extending off of the template, then the incorporation of the next nucleotide occurs at some rate. But if you've made a mistake, so you've already made a mistake, the extension or the further protection of the new sequence is slow. And this has been observed for a variety of protein polymerases, and the ratio between these rate constants is anywhere from 10 to 10 to the 7, depending on the enzyme and the particular base pair involved. The second feature that we want to include is the fact that if you make a copy of something using a template, you have to separate the copy from the template in order to continue replication. So in other words, we need to separate the strands. If we're talking about an RNA or nucleic acid type of system, we need to, for example, heat up the system in order to break the strands apart. And this is a problem for prebiotic chemistry, which doesn't have the protein machinery that modern organisms do. So I can assume that the strands separate as needed, but we're going to sort of put this in explicitly into the model. So here's our scenario of what's happening. We've got replication and strand separation of the master sequence, perfect copies. We've also got replication and the incorporation of a mistake followed by strand separation. So we've got this fragment. And the same two processes happen for mutant sequences as well at a reduced rate. And then these fragments can then pair up with any template after the next, say, thermocycle comes along, and at some slow rate, marked zeta here, will finish extending. So if you solve this, I won't go into the details of this, but if you solve the model for the error threshold using the differential equations that you derive from those chemical equations, then you plot the maximum genome length under the prebiotic conditions divided by the original error threshold, then you see that the first thing to notice is that the error threshold or the ratio of these two lengths is always greater than 1. So including these mechanistic details always either increases or keeps the same the maximum length of the genome. And the second important point is that at low fitnesses, this effect can be really quite large. So you can get, orders of magnitude kind of increases in the genome length in principle. So the important thing maybe to ask yourself is what are the relevant mutation rate and stalling factors. So we've been looking at a couple of systems to try to understand what these numbers would have been, and the system I'll tell you about is the RNA polymerase ribozyme. So this was a ribozyme which was developed in Dave Bartel's lab, which essentially copies a piece of RNA. So it's kind of a step toward a self-replicating RNA system. So the mutation rate of this enzyme is known to be around 3%. And we determined the stalling factors. So this work was done by Justin Achita and Jack Shostak's lab. And what he found, what he did was to take a template sequence and all possible matched and mismatched termini. So he determined the extension rates off of matches and off of mismatches. Just a couple of examples here. You can see that primer extension for a matched situation occurs on the timescale of minutes, whereas the timescale for a mismatch situation is hours. So we collected all this data, averaged it together in the appropriate way by weighting according to the misincorporation rates, and came up with a overall stalling factor of around 10. So this means that the ratio of the rate of stalling from extension from a mutant compared to a exact copy of the master sequence would be around a factor of 10. So this translates into an increase in the genome length by 35%, and we want to sort of see what these sorts of numbers are for different prebiotic systems. Okay, so the second thing I want to talk about is why does complexity increase over time, and in particular just focusing on this transition and the information there. So first of all, what sort of sequences do you start out with? If you think about prebiotic chemistry as being sort of monkeys at a typewriter that are producing a whole bunch of sequences, these monkeys are actually not typing totally randomly. And they're probably tending to make simple sequences. And there are a couple of reasons for this. The first reason is that not all of the starting materials, not all of the monomers, are going to be present in exactly equal concentrations. So this is going to bias the composition of the final sequence. And the reason for that is because they're made by different synthetic routes, and you can actually see evidence of this if you look at meteorites that contain nucleobases. So those meteorites tend to lack cytosine, even though other nucleobases are present. And the other general reason why you might tend to make simple sequences is that the rates, the different monomers have different rates of reaction. And for example, in Jim Ferris's lab, they've looked at polymerization, which is catalyzed by mineral clays. And they found that the pyrimidines are more reactive than purines under their polymerization conditions. And this difference propagates as the polymer increases in length. So if you look at pentamers, 5-mers, they had 512 possible pentamer sequences, and of those theoretically possible pentamers, they only observed 4 of them. So this is obviously a large decrease in the possible complexity of the pool. So, so this raises the question, where did complex sequences come from? So the synthetic mechanism I'm going to talk about is template-directed synthesis. The basic idea here is that if you have a template, then the presence of the template catalyzes the bond-forming reaction between two complementary fragments, thus creating the complement. And the idea is that the presence of the template increases the local concentration of the reactive ends. And this is a pretty general phenomenon that's been exploited to great utility in organic chemistry, and in principle would apply to any templating polymer. So just to give you some examples of this type of chemistry for self-replicating systems, oligonucleotides have been shown in a variety of different systems, different activation chemistries, et cetera, to exhibit this phenomenon. There's a very interesting example in peptide chemistry by Reza Ghadiri's group, kind of a molecular ecology of peptides. And even for small molecules, you can engineer a system which shows this effect. Okay, so using that as the backdrop for what the synthetic mechanism is, let's ask how does the complexity of the sequence influence this mechanism? So if we think about a repetitive sequence, this is a very simple sequence with low Shannon entropy. If we consider fragments that are made that could make up the sequence, for example, they'd be produced by breakdown of the polymer, then there are two things that could happen through template-directed synthesis. One is that you simply regenerate your original sequence, since you could pair these fragments up again with the template. The other thing that could happen in this situation is that you could swap the fragments because this group of C's could bind with this group of G's and vice versa, and then you would end up making errors. But instead of a simple sequence, let's say we have a complex sequence, for example, kind of a random sequence. If we have the same situation, fragments of these sequences, we could also, through template-directed synthesis, regenerate the sequence. And if we ask, could these fragments swap partners, the answer is no, because of the specificity in base pairing. So, so we hypothesized that if the sequence is more complex, it will be worse at regeneration. So the first thing we did was, before sort of jumping into experiments, we did some computer simulations to examine this effect. Basically, a very crude thing, made a whole bunch of sequences, broke them up into pieces, annealed them, ligated them together using templated synthesis, and asked what the products are. And if you do this for a whole bunch of different sequences, and you plot the Shannon entropy of the sequence against the fraction regenerated, you can see that there's a pretty strong dependence of the regeneration on the entropy of the sequence. So this suggests that the more complex the sequence is, the more fit it is during this process. And we've also been doing some experiments along this line, and I'll just show you two examples. One is an extremely low complexity sequence. It's essentially as low as the complexity can get. It's a homopolymer. And on the right side, we have a high complexity sequence, which was a randomly generated sequence. So looking at the homopolymer, if we take a look at the ligation reaction, we're expecting a product of length 40. And you can see that there are a whole bunch of non-specific products. And if you quantitate how much ended up in the 40 mer band, it's around 3%. If you look at the high complexity sequence, it's a very different story. The ligation is very specific, and you get almost quantitative yield of the correct length polymer. Okay, so the sort of take-home message from this is that the information entropy correlates with fitness under this sort of prebiotic replication cycle. And if you have a low complexity sequence, this process ends up producing a whole bunch of different fragments. If you have a high complexity sequence, this process will inevitably sort of degrade some of the information, but will also preserve some of the information. So if you also had a mechanism for a replication, then you can imagine the reproductive ratio being greater than 1. So what I told you about was for sequences in isolation. And in reality, we would have different sequences in competition with each other. So we also wanted to examine what would happen if we put different sequences in the same tube. And in terms of simulations, we basically just carry out the same algorithm. If you just kind of ask yourself, what do you expect? Well, if the sequences have no overlapping areas, in other words, they can't cross-hybridize with each other, then you don't expect the fitness of either sequence to be affected by the fact that you've mixed them together. But if there are some opportunities for cross-hybridization, so for example, in this pair you can see many opportunities for cross-hybridization, then you do expect these sequences to be able to cross-hybridize and produce different products. So if you, I haven't shown it here, but if you do the simulation with totally orthogonal sequences, then you don't see any change in fitness. But if you do it with related sequences, then they do interfere with each other. So if you look at the fitness of each sequence alone and compare it to its fitness in competition, then in both cases it's decreased. And if you do this for a whole bunch of different sequences, so here we're plotting the relatedness of the sequence, the probability of cross-hybridization, against the fitness of the sequence in a mixture divided by the fitness of the sequence in isolation, then you can see that there's a negative relationship, just like we expect. So I think that the message from this is that the more diverse the pool is, then the more fit the pool is. And these are kind of, these two sort of results are flip sides of the same coin. If you ask yourself in an isolated pool what sequence does better, it's the sequence that has more internal diversity. If you ask yourself in a mixed pool which pools do better, it's the sequences that have a greater overall diversity or sort of external diversity. So just to summarize, we've looked at how the mechanism of synthesis influences the informational properties of the polymers. First of all, if we incorporate kind of a realistic prebiotic replication cycle, then that appears to postpone the error threshold, and we're still working on exactly how much by how much that's postponed. And the second point is that the mechanism of template-directed synthesis, which would be a mechanism for recovering sequences after degradation, this mechanism favors complexity and diversity in the sequences. So finally, I'd like to thank all the people who are involved. The members of my lab who are involved in the error threshold project are Sudha Rajamani, who is working on non-enzymatic synthesis. And the people who are our collaborators are Jack Shostak, who is also my thesis advisor, and Justin Achita and Doug Trico from the Shostak lab. Martin Novak and Tibor Antol worked on the kinetic modeling for the error threshold. For the complexity project, this was really pushed forward by two undergraduates, Isaac Joseph and Kevin Liu, and I'm very grateful for advice from David Liu and Eugene Shaknovich, and to the Bauer Fellows Program for financial support. Thanks.
Time for questions.
So, Irene, could you go back to the first half of the talk? I'm a little confused about the model and the consequence. So, if I understood you correctly, you were saying that you assume that replication is slower after an error, or extension is slower after an error, and somehow that gave you longer templates. How did that get you to longer templates?
Oh, let's see. So I guess the idea is, we're not, in this model, we don't have a mechanism for making templates longer, but we're just asking for templates of different length, would they survive or not survive?
So what was the graph that the colorful rainbow one? Yeah, that
Yeah.
one. So at high error rates, you said you got long templates. What does that mean? What does this graph mean?
Oh, so what's plotted here is the ratio of the new maximum versus the old maximum. So, for example, if you're down here at 1, then incorporating this effect makes no difference. And if you're, say, up
so
here,
you can faithfully replicate longer templates with your model than with the simpler one.
Right, exactly.
Got it.
Yeah.
Other questions? I have a question. Is that so? What you meant is that complexity is better, diversified sequence is better because it fits most. Right?
Yeah.
So is it the sequence length dependent phenomenon, or whether by the time your sequence is increased to certain lengths, you don't, in a sense, I will see whether it's the correlation is actually more like once you increase to certain sequence, you don't see that strong contrast anymore.
Oh, okay, yeah. So for those experiments and simulations, we use sequences of a fixed length. So we take that out of the calculation of it's not varying in the calculation of how complex the sequence is. So for complexity, we're measuring it just by the internal diversity of a sequence of a fixed length.
Okay.
Yeah.
First, this is a very interesting talk. I have two questions. First is technical question, and when you do the replication, what kind of polymerase are you using? Are you using a proofreading polymerase?
Okay. Oh, so are you talking about the error threshold or about? Oh, okay.
Yes.
There's no proofreading in the enzyme. It's a ribozyme, and the error rate is known for that ribozyme, but there's no sort of additional proofreading step.
Right, and that's a follow-up question. And we know that actually the polymerase actually used in the cell is the error rate is very low. It's 1 out of 10 million, right? And do you think through evolution, the error rate of polymerase is increasing? It's decreasing, so that maybe in the early days and the error is very high and gradually going down. And how that will fit into your model?
Yeah, I definitely think that is probably true, that the prebiotic mechanisms of replication were low fidelity, especially compared to what we have now, which has many mechanisms for ensuring fidelity. So it's probably true that, and even if you just look at the mutation rate for this in vitro evolved ribozyme, it's 3%, which is pretty high. So it's probably true that the prebiotic mechanisms are really high error rate and then evolved to become lower. And what direction the evolution should be in, it sort of depends on exactly what the situation is. So we know that for example, higher error rates for some viruses might be beneficial, and some viruses live at the error threshold. But on the other hand, very, very large genomes and more sort of stable organisms would be harmed by having such a high error rate. So different organisms probably have different selective pressures.
Any other questions? I have one more question.
Yeah.
Okay, I went to Ari Winfrey's talk, or he's here. So he's from crystal to life, right? And in his models, actually he can comment on it. But the diversity is bad, right? The error rate is not good for his crystals, right? But why yours is? Can you comment on that? Because it's a similar materials, right? But from a crystal to grown, that you cannot tolerate any errors in terms of segments.
Oh, maybe the question is what would the error threshold be for?
I think the topic here and yours actually there's some similarities.
Yeah, they're very, very closely related. In fact, I think the same kind of issue would come up. I mean, if you're replicating information by crystal growth or by direct templating, in fact, one can think of them as the same thing. A crystal is a templated growth process that doesn't dissociate. It just keeps templating again and again to build up multiple layers. And so you have the same problem that as errors accumulate, at some point you can't propagate a very long sequence.
But in the crystal case, do you see the heterogeneous crystal segments versus more homogeneous segment? Did you see is better? Is in the sense that the heterogeneous one is better than the homogeneous one, as she has seen here.
So I guess the difference in our model, the crystal growth occurs one monomer at a time, rather than the model that was considered here, where there was a fragmentation and then a binding process. So I guess that was the key to this model in some of the cases. So do you agree with it?
Yeah. Yeah, monomer extension, I'm not sure I can see how if we were building up from monomers, whether complexity would have any effect.
Okay. Any more questions? Okay, that one is.
I was just a little confused at the last. I mean, you're talking about the relatedness of the one sequence to others, right?
Yeah.
To avoid the mismatch or some kind of things that the sequence, master sequence, should be isolated from others, right? In a
Right.
sense, right?
Right.
But at the same time, of course, there are always point mutations.
Right.
And, how and the total or average fitness around him
Yeah.
is going to be trouble, right? So how can you deal with that?
Yeah, I think that's a really interesting question, and we're going to try to, get into that. But my kind of intuition is that if you start to make point mutants, a couple of things can happen. If the point mutants if there are enough of them, that you depress the fitness of the whole pool, then that can sort of lead to a kind of error catastrophe. But if the mutation rate is really fast, then you could mutate to the point that you would not have any relatedness between the two sequences. So, and then that would increase the fitness of the pool. So it's an interesting balance.
Or how you can reach such kind of the isolated sequence from the any arbitrary point in the sequence space. It's kind of isolated, so it means that it's small basing in a sense that it's kind of hard to reach that kind of isolated guy from the bias point.
Right. I think what we need to do is to test this out using starting from a random pool and then see what evolves, basically. Yeah.
Okay, I think that concludes this section. Please join me to thank Irene again.